Hello, welcome to another edition of the CNA's Opus. My name is Rich. I'm a certified nursing assistant, and today I want to talk about Burkitt's lymphoma. Lymphoma is the cancer of the lymph nodes, and Burkitt's lymphoma is the cancer of the B-cell lymphocytes. So, uh, just starting out with the structure of the lymph node has these incoming lymph vessels which go into this outer region called the marginal sinus. And the lymph sits in this marginal sinus here. And in between there, it has kind of like this filtration system. It has these B cell follicles, which hold the B cells right there. And in between there, kind of like in free form, in between there, they have the T cells, which are both white blood cells that are part of your immune system. And then within that region, there's something called the cortex and the parietal cortex and the medulla. And then from the medulla, it has the outgoing lymph vessel. So the lymph that's sitting in the marginal sinus there, it wants to get from there to the medulla and out. But in between there, it has to get through all those white blood cells that are kind of acting as this filtration system. And the reason being is that if the lymph comes in and has anything that's foreign, bacteria or virus, parasite, whatever it is, anything that's foreign, it has the white blood cells there that'll pick it up, detect it, and set off an immune response while it's trying to get through. So that's the reason for that. With Burkitt's lymphoma, the B cells again are in these B cell follicles right there. What happens what first happens is a gain of function mutation, and the gain of function mutation is due to a translocation. So it's a reciprocal translocation, and if I could just show it right here, this is chromatin 8 and this is chromatin 14, and in here I have remember you have 23 pairs of chromosomes which make 46. So one of the pairs is going to be the chromatin, which is going to have the allele, one of the alleles, or variation of the genes from your mother or from your father. So this one in here, that's in blue, that's one of the alleles, either from your mother or from your father. And that in red right there is the promoter region. And the promoter region up here is for c-Myc, and c-Myc is a mitogen. It drives cell growth. It drives cell proliferation. But the promoter region for c-Myc, it is very slow acting. RNA polymerase complex 2 gets to it very slowly, and then it transcribes c-Myc at a slow, steady pace for cell growth and cell division in normal cells. Down here, since these are B cells, they're white blood cells, one of its jobs is to make the antibodies. And right here on chromatin 14, this is an allele called IgH, or immunoglobulin heavy chain. And one of the jobs of the B cells is to make the antibodies, which are made up of immunoglobulin. And this is for the heavy chain. The heavy chain down here in the antibody, if you see this V shape, the big V right there, that's the heavy chain. That's the heavy chain. So that's what that's making right there to make the antibodies, to bind to the antigens. And the antibodies right here, the heavy chain is held together by these disulfide bonds, or SS bonds. And the heavy chain to the light chains are again held together by these disulfide bonds, or SS bonds, or SS bridges, or a bunch of different names for it. But what it's making here is for the heavy chains, and this promoter region right here is acting very quickly. So RNA polymerase complex 2 is getting to this constantly. Boom, 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 quick, 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 and making the messenger RNA right here at this allele for the immunoglobulin heavy chain very quickly to make the heavy chains for the antibodies. So when this reciprocal translocation occurs, if I could just draw this down right here, when this reciprocal translocation occurs here and here, I don't want to get too sloppy, but I know everyone's busy. See, when it makes, it'll make it cut right. It's a translocation, so there's a break right here, and there's a break right here, and then they switch. So now the c-Myc is next to a promoter region that's very, very active. So the c-Myc, the messenger RNA for c-Myc, is constantly being made and transcribed right there. So there's a lot of c-Myc being produced to push cell proliferation. And we would call that an oncogene. Up here, it's a pro-oncogene. It's just for normal cell growth. But once that mutation occurs, it becomes an oncogene. It's driving cell growth at a faster rate than it should be, and that's one of the hallmarks of cancer. Okay, because it's next to a promoter region that has a very high activity. What happens to the immunoglobulin heavy chain allele goes up to 8, and that has a very slow acting promoter region, and that really doesn't drive towards cancer. If anything, it's going to drive towards a lower immune response, because now it's going to make the immunoglobulin heavy chains at a much slower rate. Therefore, there's going to be less antibodies. So it's going to be kind of a partial loss of function of an immune response, but not a loss of function or gain of function that's going to drive cellular proliferation. This one, however, does when c-Myc goes to this one on 14, and it's the messenger RNA for c-Myc is made at a very, very fast rate because of that translocation. And then what happens is the B cells in the B cell follicles, they start to grow, and you can often see like the lymph nodes swollen on the neck or around the mouth of the lymph nodes. That one mutation, that one gain of function mutation, is not enough to cause cancer. It usually takes about 3 to 15 mutations to cause cancer, but that one sets it off. It starts it off. It grows fast, and every daughter cell is going to have that same mutation. And because it's going so fast, and replication is happening so fast, there's there's much more room for error. It's going so quickly. So then another mutation is picked up, and then all those daughter cells have those two same mutations, and then cell grow, fast, and another mutation is picked up, and then that group of clone of cells right there will have those 3 mutations. All those daughter cells will have those 3 mutations. All the same mutations, all the daughter cells, until it gets all the hallmarks of cancer. And that's when and usually is around 3 to 15. But that's the start of it right there, is that one gain of function. Another thing that I find interesting here is that the immunoglobulin domains that are found in a lot of receptor tyrosine kinases. And I use vascular endothelial growth factor receptor 2 as an example, because all these there's 7 immunoglobulin domains found in it, which means they have an amino acid sequence that's consistent There's a segmentation that's the same as a domain, not as the entire thing. It's not, the entire thing is not the same. It just has an amino acid sequence that's conserved. That's the same. There's a segmentation. It's the same as what's found in immunoglobulin. Is the same as an amino acid sequence that's conserved, that's found in receptor tyrosine kinase. And that's in a lot of them, not just in vascular endothelial growth factor receptor 2, but in a lot of them they have the immunoglobulin domains, because it's an amino acid sequence that's conserved, that's the same that's found in immunoglobulin, which immunoglobulin is what makes up the antibodies. So if you just make it full circle right there, receptor tyrosine kinase to immunoglobulin, the antibodies, kind of makes like this dogma of a full circle. If you just want to like connect the dots, which I like doing, that's an interesting way of solving puzzles. Anyway, that's it on Burkitt's lymphoma. I hope I didn't leave anything out. And that's it. Rich, CNA. See you later.